All right, couple quick announcements as we get started here. One, I am going to be handing out exam number 4 here in a second, due on Friday. So that is something that's going to be your main focus this week, especially because for tomorrow's lab, it's optional. So if you guys don't want to have to come in tomorrow, you do not have to come in tomorrow. If you want to come in, though, I will be here as normal, and I'll be here if you guys need to be working some on your unknowns projects, on your research projects. I usually do set aside several of these lab periods here before Thanksgiving, after Thanksgiving, just because I know because of these projects that are associated with it. So just a reminder, this week and only part of next week is available to finish up research projects. So if you're still working on those needs to be getting finished up. The research project presentations then are going to be on the Tuesday after Thanksgiving break, which is basically 2 weeks. Okay, 2 weeks and a day from now. So that's where we're going to have the research presentations. Again, those are going to be oral presentations. They're going to be worth 50 points over your research projects, and you're going to be just presenting those to the class that particular day. And just like I've done in the past, I'll probably just do a random drawing to decide what the order of presentations is going to be. Okay. But we're going to do all the presentations on the Tuesday after Thanksgiving break. So we have exam number 4 due Friday. We're gonna have the research presentations after break. Tomorrow's lab is optional, but I'll be here to help with unknowns project material with then research projects. Questions about that then? All right. Otherwise, today we're going to keep going through lecture. And we talked a little bit about transcription last lecture. Started talking about prokaryotic translation, and that's where we're going to pick back up. So last lecture in particular, I left off talking about the genetic code. The genetic code is, remember, the flow of information inside cells is DNA to RNA to protein, when we're talking about the flow of information inside of cells, or the central dogma of biology as it's sometimes referred to as. With that, the codons are a triplet of nucleotides that codes for one amino acid. So for instance, AAU is a codon in the messenger RNA, and this would end up coding for putting an asparagine amino acid into that. But we can look at this. So it's a 3 bases that equal 1 amino acid. You got specific sequences for specific amino acids. You also have specific sequences for then start and stop of the translational process, which we're going to talk more about those in just a second here. Remember, tRNAs also facilitate translating nucleotide languages. So there's going to be a codon in the messenger RNA. This will complementary base pair with an anticodon in the tRNA. And that anticodon is always paired up in that this particular tRNA is always paired up with the exact same amino acid every time, in this case, phenylalanine. So what this does, tRNAs translate a nucleotide sequence into and then a nucleotide language into then an amino acid language. Now, the messenger RNA, the tRNAs, all of this then comes together inside of ribosomes. And ribosomes are going to be the site of protein synthesis for cells. And it's not just one ribosome. There's thousands of ribosomes per a individual bacterial cell. There's thousands and thousands of these per even eukaryotic cells, probably more than just a few thousands. Ribosomes in prokaryotes are big enzyme complexes, but often they're broken down into 2 different main subunit parts, what's called the 50S subunit versus the 30S subunit. And these are not, these are big enzyme complexes. The 30S subunit is composed of 21 different proteins, plus the 16S rRNA molecule, a ribosomal RNA. And we're going to talk about the role of 16S ribosomal RNA here in a second. The 50S subunit's even bigger. It's about 31 different protein enzymes make up the 50S subunit, plus the 5S rRNA and the 23S rRNA. So a couple different ribosomal RNAs are then in that 50S subunit. So there's a lot of different enzymes and some ribosomal RNA molecules that are forming these kind of big superstructures inside the cell, and they're all cooperating towards a particular function. Now, these 30 and 50S, they start off apart, but then they come together around a messenger RNA. The messenger RNA kind of gets sandwiched in between them. And when they come together, that 50S subunit plus the 30S subunit then forms what is called a 70S ribosome. And I know that does not add up. This is not a math equation. The S just stands for sucrose gradient. It's a measure of the mass of these things, but masses are measured on a logarithmic scale as opposed to a linear scale. So even though this is a 50S sucrose gradient mass and this is a 30S sucrose gradient mass, when they come together, they only equal a 70S because it's a logarithmic scale by which you measure the mass in what are called sucrose gradients. It's a really old way. Sucrose gradients was a really old way to, and a centrifugation technique to separate different masses of cellular components. So this we can always come out in what is called a 70S fraction. Okay. Now, ribosomes are, again, where translation or protein synthesis take place. And what they do is their job is to help the coupling of codons in messenger RNAs to anticodons in tRNAs and transfer RNAs. So they make sure that there is a proper codon-anticodon pairing. And then if that pairing is kind of spell checked, validated, it then joins together the amino acids. It lines the amino acids up and then joins them together via formation of peptide bonds. All right, so what we're going to do is talk about what happens during this process. How do they make sure the mRNAs and tRNAs are lined up correctly? How do they join together amino acids to build a protein via the formation of these peptide bonds then? So we're going to take a look at this like we have the other sequences. We're going to focus on start, what happens in the middle, and then how you terminate. So initiation, elongation, termination. How do you start this process? So here I have a gene, DNA. Off of it, you're making a messenger RNA transcript. Okay. In that messenger RNA transcript, initiation is going to start off, ribosomes are first going to come onto here with the 30S subunit coming in first. And so the 30S subunit is going to bind to the messenger RNA. It's going to bind at the beginning of the coding sequence. And there's going to be several enzymes called initiation factors, like initiation factor 1, initiation factor 2, initiation factor 3, IF1, IF2, IF3. It's going to use some energy in the form of GTP to help make sure that it gets all loaded in on the right spot. But the question is also, how does the 30S subunit know where to bind to then start this process? So how does the 30S subunit know where the start of translation is? Anybody have any ideas? Okay, AUG is what we also call sometimes what's the name for an AUG? Codes for methionine, but it's also the start codon. So that's going to be involved. So yeah, the start codon is going to be part of this. But am I marking anything else looks kind of weird in there? Or is there, didn't I have something else right in front of the start codon? Well, there's something that's going on here. Let's say you have a messenger RNA. Do you guys happen to remember from the genetic code how many codons code for methionine? One. Yeah, just one. There's actually only one codon, the AUG codon that codes for methionine. Now, you might have, though, multiple methionines in a protein. So let's just say you have a messenger RNA. That messenger RNA, it may need to start translation here. This will be the translation start, for instance. And you're going to have an AUG codon at the beginning. But you might have another AUG codon here. You might have another AUG codon over here. You might have another AUG codon over here. And it isn't until over here that maybe you then do something like transcriptional termination. Okay. So the question is, which one is the right AUG to be a start codon, a true start codon that says start translation over here? Because if you could bind, the ribosome binds to this AUG codon, then it goes all the way over to here. Yeah, you get the entire protein made, for instance. But what happens if it binds here? Well, then it starts making the protein from this point on. And guess what? You're missing all of these parts of the protein. Does that make sense, everybody? So if there's multiple AUG start codons, and that means in a particular protein's code, amino acid sequence, which one is actually the start codon? Which one's the one that you have as the beginning of the protein then? So when ribosomes are looking for, it is a start codon, but it's more than just a start codon. So how does that 30S subunit know where to bind and find the right place to bind? It is an AUG start codon, so that is involved, but it has to then be preceded by a type of ribosomal binding site called a Shine-Dalgarno sequence then. So it has to be, again, a combination of, yes, there's an AUG start codon, but there also has to be the Shine-Dalgarno sequence in front of it. And it's usually about 10 base pairs in front of it in the RNA. So again, this is going to be part of the messenger RNA. This portion right here, though, this is going to be the first base pair that goes into a protein. So this portion right here and on is probably what we call the coding sequence. Codes for the actual protein sequence. This right here is going to be a untranslated region. That means it does not directly code for a protein, but those untranslated regions can still be important. For instance, the Shine-Dalgarno sequence is in the untranslated region of the messenger RNA molecule. So these are all messenger RNA sequences. That untranslated region is part of it. If you don't have the Shine-Dalgarno sequence, do you get translation? No. You need the combination of an AUG with a Shine-Dalgarno sequence in front of it. These are, again, still RNA sequences. They're part of the messenger RNA molecule, is what I mean by that. But the ribosome recognizes that start codon with the Shine-Dalgarno sequence in front of it. And that triggers the start of translation. Questions about that? Okay. Technically, what actually happens is, again, the Shine-Dalgarno sequence is often a sequence like I think this is a fairly common Shine-Dalgarno sequence, for instance, in E. coli. Again, you're going to have some random base pairs. And then you have the AUG start codon. This is a little roughly what it looks like. The Shine-Dalgarno sequences, different species are going to use different Shine-Dalgarno sequences. So this is roughly what it kind of looks like. These are X's are just random base pairs. Again, this is all in the messenger RNA. But the 30S subunit is going to come in and recognize that and bind to it then. And it recognizes it actually because a little portion of the 16S RNA sticks out of it. So this is going to be my 16S RNA sticking out of my 30S subunit here. And what it does is it complementary base pairs with the Shine-Dalgarno sequence. It helps it line up correctly then. Questions about that?
So what happens with like the 10 in between, like that just not found at all?
It's just not, yeah, it's that sequence isn't particularly important.
Okay.
It's just spacer. Okay. Once that is lined up, it's always lined up very accurately on what is called the appropriate reading frame. So it always lines it up on that start codon. And then the reading frame is any start codon where you start translation. And then it's all the way, the reading frame or coding sequence is until you reach what is called an in-frame stop codon, one of the 3 stop codons of UAA, UAG, or UGA. And so what ends up happening is if you read it correctly, you get, for instance, a start codon, then you read it from that point on 3 bases at a time. Okay, the ribosome reads this 3, then slides over to the next 3, then slides over to the next 3, then slides over to the next 3. It always reads it 3, slides over 3, reads the next 3. Slides over 3, reads the next 3. Slides over 3, reads the next 3. Does that make sense already? If you slide over the wrong number of base pairs, say you slide over to the right by accident, then you actually are reading the wrong reading frame. So in this case, if you actually slipped -1, now instead of reading ACA here, you slip to the side a little bit, you're reading AAC. That's asparagine instead of threonine, then isoleucine instead of tyrosine. So if you slide over incorrectly, either 1 base pair to the left or 1 base pair to the right, you get different proteins made. So the ribosome has to be lined up very precisely with that combination of the Shine-Dalgarno sequence and the start codon, or you get the wrong proteins made. Questions about that? All right. After that happens, you've got 30S subunit now attached, recognizing the Shine-Dalgarno sequence, recognizing the start codon. What will then happen is there's an initiation factor IF3. This prevents the association of the 50S subunit until 30S subunit and initiation factor 2 recruits a tRNA carrying methionine. By the way, what will be the anticodon on my tRNA carrying methionine here? What would be the anticodon? It's a tRNA, it's an RNA, so it's going to be U. AC. Yep. Sorry, Mitch, I couldn't hear you. I think you said it. I think you said it. All right, so it's going to be a tRNA with the UAC anticodon carrying methionine. That comes in, interacts with the start codon. This requires an input of energy, and the energy in this case is going to be GTP. GTP is going to be this guanine triphosphate, and hydrolysis of that is going to be the source of free energy that drives this process of translation. Once that's brought in, that bumps initiation factor 3, IF3 off, and allows then 50S subunit to come in. So once this is in there, it'll take that initiation factor 3 comes off and allows 50S subunit to associate. And that makes what's called the 70S initiation complex. So that's how you start up translation. That's how you first load ribosomes onto a messenger RNA transcript molecule. What we're going to talk about now is what more happens during elongation, and take a closer look at some of the different things in then the ribosome. So what I have here is just to give you guys a little bit of different things. Here again is, I've flipped it a little bit. This is the 30S subunit. This is the 50S subunit. In red, I've got my messenger RNA molecule. These kind of, I don't know, what color is that? Teal? Are the tRNAs. This is the anticodon on the tRNA versus the amino acids that it's carrying. So purple are amino acids. What I'm showing you here is in the middle of elongation, there is already a 1, 2, 3, 4 amino acid long protein being made. This is the next incoming tRNA carrying the next incoming amino acid coming in. If we look inside of ribosomes, they're said to have 3 different kind of main positions inside the ribosomes, what's called the E site, the P site, and the A site. E site stands for exit site, and we'll talk about what that does here in a second. The P site is what is called the peptide site. It's the main site where peptide bond formation is going to take place. And the A site is called the acceptor site because this is where incoming tRNAs are going to come into that. So if we're looking what's going to happen, here we have an empty A site to start off with. We have a tRNA that's carrying the polypeptide chain that we are building then. So here's the growing polypeptide chain. Here's it's attached to a tRNA in the P site. The A site's empty, but here's a tRNA that's going to come in to that A site. What's going to happen is there is a enzyme called elongation factor TU, or EFTU for short. This uses energy in the form of GTP to kind of make sure that this incoming tRNA comes into the ribosome correctly and make sure it's oriented properly. It even does a little bit of proofreading or spell check to make sure that there is a proper codon, anti-codon recognition. Then. So it helps load that incoming tRNA carrying that charged tRNA, carrying an amino acid into that A site of the ribosome and make sure it's lined up correctly. This is then going to result in the amino group of the amino acid on that charged tRNA in the A site interacts with the carboxyl group of the amino acid, the last amino acid coming off the tRNA in the P site. So again, you're going to get, the carboxyl group of this guy interacts with the amino group of this guy. And so as they interact, they're going to form a peptide bond. What that does is this is catalyzed by 23S rRNA. Remember, most enzymes are proteins. Remember, there's always a few enzymes that are RNA molecules. This is an example where an RNA molecule is functioning as an enzyme. 23S rRNA is actually the main enzyme that facilitates peptide bond formation. This carboxyl group interacts with the amino group. And what that does is it attaches on that growing polypeptide chain onto the tRNA coming off the A site now. And you've grown that polypeptide chain by another amino acid. Questions about that? What you've got now, though, is you form that peptide bond. You've now got an uncharged tRNA in the P site. This is an uncharged tRNA is one that's not carrying an amino acid. And you've passed the growing polypeptide chain onto the tRNA in the A site. This is going to trigger translocation. Another elongation factor called EFG is going to come in. It's going to spin another GTP molecule as a source of free energy, and it causes the ribosome to slide over 3 base pairs, 3 nucleotide positions. So this, this is going to slide to here. This is going to slide to here. Going to slide over by 3 nucleotides. This moves that uncharged tRNA from the P site into the E site, the exit site, and then it's going to exit out of the ribosome. It's also going to move this charged tRNA carrying the growing polypeptide chain from the A site into the P site. And then that is going to create an opening in the A site. And then you're just going to repeat that entire process again during elongation. Questions about that? How many GTPs are you spending per amino acid you're joining together? 2. One right here, and one right there. So for every amino acid you're joining together, you're spending 2 GTP molecules, or in essence, 2 ATP equivalents. That means building proteins is pretty expensive. Questions about that? All right, terminating transcription then. I'm sorry, translation, terminating translation. I always do that. I always mix those two constantly. Don't do that on exams. All right, anyways. When you terminate translation, this is that ribosome's just going to keep doing this process over and over and over and over and over until it reaches a stop codon. And that's when termination is going to happen. So with a ribosome, when it reaches a stop codon, there is no tRNAs that code for a stop codon. There's no tRNA that corresponds to that stop codon sequence. So the ribosome is going to pause when it reaches a stop codon. But then some different enzymes called release factors are going to come in. There's 3 different release factors: release factor 1, release factor 2, and release factor 3. Why are there 3 different release factors? Because there's 3 different stop codons. Each release factor corresponds to a specific stop codon. Release factors come in, and then what they do is they almost act like a wedge. They basically are going to tear apart the 30 and 50S subunits. So the 30 and the 50S subunits will come apart, the ribosome will come apart, and they also cause the release of the peptide chain from whatever tRNA it's attached to. And then you've made a protein. Questions about that? Now, there's a couple other things to keep in mind, especially when it comes to prokaryotic translation. First off, it typically prokaryotic translation is typically going to be taking place as big complexes called polysomes. And polysomes, this is where you have translation of a single messenger RNA by several ribosomes simultaneously. So this is not meant to be like a timed over time image. This is supposed to be a static image, an instantaneous snapshot. But what it is, it's supposed to be one messenger RNA is being bound by 1, 2, 3, 4 different ribosomes. And this would be what an example of a polysome. It's one messenger RNA being bound by several ribosomes at the same time. But maybe this ribosome has slid all the way around the messenger RNA and it's finished making its polypeptide and it's about to dissociate. This ribosome is maybe 3/4 of the way, so it's made part of the protein, but not all of it. This is maybe half, so it's made some of the protein, but maybe only half the protein off of this messenger RNA transcript, and maybe this one's only 1/4 of the way through. So you have these different ribosomes all making protein at the same time off of a single messenger RNA transcript, though. Why do this? Again, the same thing here. You're seeing a messenger RNA, but it's got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, for instance, ribosomes all bound here at the same time off of that messenger RNA. What's the benefit of having what are called polysomes? Yep. Faster. And what that really just boils down to is more protein made per unit of time. And that's especially beneficial if you're making proteins the cell needs a lot of. which is many different kinds of proteins, actually. The other thing to keep in mind in prokaryotes is that in prokaryotes, but not eukaryotes, transcription and translation are coupled. So I say the coupling of transcription and translation, this does take place in prokaryotes, but does not in eukaryotes. So eukaryotes do not have coupled transcription translation. And what I mean by coupled transcription translation is that in prokaryotes, translation will start before transcription even makes a final finished messenger RNA. So the messenger RNA molecule is only half made, for instance, and you would already be starting to make protein off of it. And it just looks a little bit like this. Again, this is an electron micrograph image showing this actually taking place in a prokaryote. But it looks like this. This is my DNA. Maybe my beginning of my gene is here. My end of my gene is, say, for instance, over here. This is RNA polymerase. It's reading the gene. This is a second copy of RNA polymerase, a third copy of RNA polymerase, a fourth different copy of RNA polymerase. Well, what happens is, right now, this gene is being copied by 4 different RNA polymerases simultaneously. Okay. And then these are the messenger RNAs being made, and these messenger RNAs are still also in the form of polysomes. So this messenger RNA is not even complete yet, and there's already 3 ribosomes bound to it and already making protein off of it. Again, this one's a little bit further along, but it's still a polysome. There's many RNA molecules, but again, that RNA polymerase is still copying the messenger RNA, and ribosomes have already bound to it and are already making protein off of it. Questions about that? Why does this happen in prokaryotes but not eukaryotes? Transcription is in the nucleus of eukaryotes, but translation is in the cytoplasm. So inside of eukaryotes, RNA polymerase, you only find it in DNA. You only find those in the nucleus, but ribosomes are always in the cytoplasm of eukaryotes. So in eukaryotes, you actually have the DNA in the nucleus, RNA polymerase binds to it, make us a messenger RNA, but that messenger RNA then has to migrate out of the nucleus into the cytoplasm where it then binds to a ribosome. You cannot couple transcription and translation together in eukaryotes because they're in 2 different compartments of the cell, 2 different places inside the cell. So you can't couple that together. But in prokaryotes, remember, transcription is in the cytoplasm, but also translation is in the cytoplasm. They don't have a nucleus. Everything's just in the cytoplasm. So they can couple this together. The RNA polymerases, the DNA, the ribosomes, they're all in the same spot, and they can be, therefore, coupled. What's beneficial about coupling? It's faster again. More protein being made per time. But the other benefit of that is it also means faster responses. Remember, gene expression is often regulated. We turn genes on and off. But say conditions change. Bacteria senses that change, changes its gene expression, but it's going to build up proteins to help it deal with that change faster. So it responds faster. Does that kind of make sense, everybody? We've talked a little bit about this a couple times already this semester. All right. Questions then about transcription or translation? What's a gene? Just say that again. Sorry, I think I missed the beginning.
Sequence that codes for something regarding.
A sequence of what? DNA. It's a sequence, a DNA sequence or a segment of the DNA that codes for a functional product, a final functional product. And yes, sometimes that final functional product is RNA, but sometimes the final functional product is a protein done instead. Okay, but it codes for some type of final functional product. So with genes, again, a segment of DNA that codes for a functional product. And we've talked about several sequences, not just the coding sequences, but also things like promoters. They're encoded in genes. Scheindel-Garneau sequences are encoded in the genes. Translational starts and stops are encoded within the genes. Sometimes things like row-independent terminators are encoded in genes then as well. Questions about that? And this is really what you're looking at, even there's some changes here, but this is often what you're looking at even with something like a eukaryotic genes, but with some differences. One thing I want to point out though, is when it comes to prokaryotes, often their genes are also organized in what are called operons. Bacterial, I should say prokaryotic genes really. Genes are often organized into operons, not always, but often are. By the way, this is something that, yes, prokaryotes have, but that no, eukaryotes typically do not. Eukaryotes typically do not use operon genetic organizations. So prokaryotic genes are often organized into operons, usually not eukaryotic genes. But an operon is when you have multiple genes being expressed from one common promoter. And this is another way to say that is genes are being co-transcribed. So what it looks like is this. This is my DNA sequences. This segment of the DNA codes for gene number 1. This segment codes for gene 2, codes for gene 3, codes for gene 4, codes for gene 5. So this segment codes for gene 5. But what you have is in front of all of this gene set, you have one promoter. And it's in front of gene number 1. And what that does, remember the promoter says, start transcription here. And so it does, it starts transcription here, but then the RNA polymerase will make a messenger RNA that goes through here, keeps going, goes through gene 2, keeps going, goes through gene 3, gene 4, gene 5. And it's only after it gets to the end of the operon that you then terminate transcription. So this is going to be, again, where you have one messenger RNA transcript, one messenger RNA molecule, but there are multiple genes, in this case 5, encoded on that single messenger RNA. Now, one important note is an operon then often contains genes of related function. So typically, whatever genes are in this operon, they're all dedicated to the same general process. So maybe, as an example, these are all genes or enzymes involved in glycolysis. So maybe gene number 1 codes for an enzyme that's in one of the steps of glycolysis. Gene number 2 codes for enzymes that's in maybe a different step of glycolysis, so on and so forth. But maybe they're all involved in glycolysis. Or maybe it's the lac operon. The lac operon is a series of 3 genes in an operon structure that then is involved in lactose catabolism, using lactose as a food source and eating it in essence. So there's a lot of different possible genes, but those genes are usually of related function. Another example of an operon is trip-operon, and trip-operon is a series of enzymes that's responsible for synthesizing the amino acid tryptophan, making tryptophan for the cell. So you can have these, but they're usually of related function. Now, while they're transcribed from a common promoter, and they're on the same messenger RNA, but each gene has its own Scheindel-Garneau sequences, translational start sequences, translational stop sequences then. So again, there's a Scheindel-Garneau and start codon for gene number 1 and a stop codon. There's a Scheindel-Garneau and start codon for gene 2 and a stop codon, so on and so forth. So what that means is that ribosomes will bind here, they'll make the protein, but then they'll stop here. And so that gene number 1 is making protein number 1. But again, a different ribosome might bind here and then make until it hits that stop codon here, and you get gene 2 expressed into protein 2, gene 3 into protein 3, gene 4 into protein 4, gene 5 into protein 5. So even though they're all in the same messenger RNA, you still get 5 different enzyme proteins made. Does that make sense, everybody? Why? Nope. In this case, it's actually not because it's faster, technically speaking. Good guess, though. It will not. It might, but that's not the main reason. And if it is, it's only going to save a tiny bit. Genes are not always on. For instance, lac operon. You might have had the talk about in different classes. It's on if lactose is present, because there's lactose is available as a food source, but the lac operon is off in the absence of lactose. Okay. Here's the thing, though, is you have to have systems to turn gene expression on and or off. And what you can do is, let's just say you have this particular operon, it needs to be on. You can have a thing that either blocks or restricts access or permits access to the promoter. What you can do is you can regulate expression here at the promoter. But if you regulate the expression at one spot, at just this promoter, you regulate the expression of all the genes in the operon simultaneously. What this does is it simplifies control of gene expression in similar gene sets. This is just a fancy way. You can have one regulatory event at this promoter controls all 5 genes simultaneously. So, and that's why the genes are organized into operons if they're of similar function. In eukaryotes, you have to turn off all the genes on and off individually. But part of the problem with eukaryotes is they don't have the same promoter sequences that prokaryotes do. So their way they regulate gene expression is different. They don't have promoter sequences like we talked about in class, and that complicates the regulation of those gene sets. Questions about that? And by the way, that does save a little bit of time and a little bit of energy. Not a ton, but it does by simplifying the genetic regulations events. By the way, ribosomes and RNAs, tRNAs are also coded in operons. RNAs, tRNAs, it might be in the DNA. You see 16S RNA. This is the gene segment for that. tRNA for methionine, 23S rRNA, 5S RNA, tRNA for alanine, tRNA for tyrosine, tRNA for glutamic acid. Okay. This is, again, one promoter expressed into a messenger RNA, and they're all on the same messenger RNA. What's the problem with that? How did you get 5 different proteins in my previous example? You had different translational starts and stops, gave you rise to 5 different enzymes. Are these expressed into proteins? Or are they functional RNAs? They're functional RNAs. So you have them all on one big long RNA molecule, but do they need to be separated at some point? Yes. So what happens is it's actually not translated by ribosomes, not translated. Instead, there's an enzyme called RNase 3 that comes in and cuts it apart. So it cuts here, 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 here. So it cuts that RNA molecule so that you get separated 16S RNAs and tRNAs and the other ribosomal RNAs, for instance. So they're enzymatically processed in order to separate them apart. Questions about that?
Well, how does that work if the enzyme needs to? Like if every stop is, oh wait, no, every stop is the same.
There's no stop codons here. This is just RNA, functional RNAs. There's no stop codons.
Oh, then how does the RNA know where to bind?
There's specific sequences. There's gonna be some specific sequences that actually fold up inside of here. But it's gonna have it recognizes a specific sequence of RNA nucleotides. And cuts at that sequence. So it's coded into it. Okay. All right, that is the end of actually exam 4 content as well. So all that stuff could be on exam 4 here. Questions about any of that? All right, I am going to keep going. So if you guys have questions, just feel free to stop by my office. We'll work through it. Okay, but the next thing we're going to talk about here is horizontal genetic transfer. So, and this is going to be exam 5 content. First off, talk about horizontal genetic transfer. I have to differentiate it from other kinds of genetic transfer. So if there's horizontal genetic transfer, what other kind of genetic transfer do you think there is? Vertical. So, first off, let's talk more about vertical gene transfer. Vertical gene transfer is reproduction. Reproduction is always involved when we're going to talk about vertical gene transfer. What is reproduction? How do you define reproduction? Make another cell, and sometimes that is ways that we do that is reproduction happens. Oh. Good. Passing genetic information to a next generation. Are there different kinds of reproduction? What kinds of reproduction are there? Main types. So there's asexual reproduction and versus then sexual reproduction. Okay. What's asexual reproduction? Yeah, it's going to be a parent making genetically identical offspring. There's two things that's part here. A parent making genetically identical offspring. It's really what we're looking at with asexual reproduction. What's sexual reproduction then instead? Yeah, two parents exchanging genetic material to make genetically varied offspring. In asexual reproduction, the offspring are what are called clones or genetic copies of the parent. But in sexual reproduction, two different parents will exchange genetic material to make offspring with a genetically unique identity or a varied identity. Which way do bacteria reproduce? Asexually. All right, and so again, binary fission, for instance, that we talked about earlier, semester, is a form of asexual reproduction, but this is also any type of reproduction equals a vertical gene transfer, either type. Vertical gene transfer is what we call reproductive events, where you pass genetic material from one offspring to a next. You guys have probably heard of like family trees. They usually go up and down or vertically, right? And so vertical gene transfer is where you pass on genetics from one generation to a next generation, or basically when you're doing reproduction. The last thing we'll talk about is just a setup here. Again, we said, do bacteria sexually reproduce? No, they don't. They reproduce asexually. With that being said, bacteria do get a little promiscuous sometimes, and they will exchange some gene segments with each other. But this is called horizontal gene transfer. This is transfer of a one or a few genes from one bacterial cell to another already existing bacterial cell. So no reproductive event, horizontal gene transfer. You got one cell, it's got some DNA inside of it. You got an already existing cell, and basically a few genes go from one cell to the other cell. Okay, not all of the genetic material, not all of the DNA, just a little bit, but it goes from one cell that already exists into another cell that already exists in some way. So it just looks like this. Genetic transfer, horizontal gene transfer requires two already cells, not a reproductive event, but some way, some DNA is going to make it from a donor cell to a recipient cell. And we're going to talk a little bit about the different ways that horizontal gene transfer happens on Wednesday. So we'll talk about transformation, conjugation, transduction.
But it can't be to a different species, right?
It can be into a different species sometimes, and often is.
What did we just talk about the other day that you said you can't pass something on to a different species because they have different.
And so it doesn't always happen.
Oh.
But it can happen.